in collaboration with Dr. Mickey Lenny, Dr. Kokendorfer at NIH, we actually chose to do a study with BCMA-targeting CAR T-cell therapy. And again, the reason to choose BCMA is because it's expressed in almost all the cases of multiple myeloma. And it also has a restricted pattern of expression on normal cells. So it's really an ideal target. And that's why we have so many BCMA targeting agents right now that are trying, that are one in the market and other ones are being developed. And so the interesting thing about this CAR T is that to reduce the risk of the immune responses against the CAR T in the recipient, in the patient, we used a novel anti-BCMA, fully human, heavy chain only binding domain, and this domain is called FHVH-33. There's also co-stimulatory domains and T-cell activation domains, and it's encoded by a retroviral vector, which is a very common way of manufacturing CAR T-cells. In this study, patients receive lymphodepletion, which is also very standard, usually with cyclophosphamide and fludarabine, and that's exactly what is being done here. And there's a planned dose escalation that had 5 levels. And then there's 12 patients already that received this therapy on 3 of the dose levels. The highest dose was 3 million CAR T cells per kilogram of body weight. The median age of the patients on the study was about 63, so they're overall younger patients, but they are very heavily pretreated. The median line of therapies is 6 lines of therapy prior to this treatment. So the very important thing is, one, the efficacy. So the efficacy seems to be very good. The overall response is very high. So 10 out of the 12 patients had at least a partial response. And even one patient achieved a partial remission 26 weeks after the infusion with a plasmacytoma decrease. So I think that this is a very active CAR T-cell therapy. And the other interesting thing in terms of possible mechanisms of resistance, that loss of BCMA expression we also observed in 2 of the patients. So some of the patients that progressed, there were 2 of the patients that progressed, but they did not have, they did not have a lot of disease in the bone marrow, only very minimal residual disease. In terms of the toxicity, this seemed like something that was tolerable. For example, there were 11 of the 12 patients had CRS, and it was from grade 1 to grade 3. However, the patients didn't, none of the patients died from CRS. There was a little bit of neurotoxicity, but it was grade 2. So there wasn't any particular intervention, and the patients, that resolved as well. And there was one patient that did die from complications of a respiratory infection that was 6 weeks after the infusion. So I think that this is initial results of a phase 1 study that actually has very good responses for actually the initial dose levels. So the number of CAR T cells infused at the dose level 3 is still relatively small compared to what the number of cells that are infused for other products. And so this is a very promising study with very high response rate. We're talking, 80-90% response rate. Even at low dose levels, it's a possibility that the response could improve even with more dose levels, and that there could be more persistence of this CAR T cells, just because they're fully humanized to avoid rejection and so on. So I think that this is a very promising study, but we have to wait and get more data, because so far only 12 patients have been treated. But it's a really good study. I mean, we collaborate. I have a lot of patients that have gotten treatment at the National Institutes of Health for BCMA CAR T-cell therapy, and it's an honor always for me to work with the folks at NIH.